Good afternoon. My name is Gabriella Cini. Welcome to my presentation on plakophilin 2 mutations and how they relate to heart defects. Let me first explain why I am interested in heart defects. This beautiful baby girl is my oldest daughter, Ida. She would be 14 years old this July. I have to say that makes me feel a little bit old. Unfortunately, she died when she was 3 days old from an undiagnosed heart defect. She was born apparently healthy. There was nothing that was picked up on the 20-week ultrasound, and she was sent home. When she was 3 days old, she stopped breathing, and we called 911, and she was rushed to the hospital. At that point, there was nothing anybody could do for her, and she passed away. She was taken up to the medical examiner's office where an autopsy was performed, and that's when they discovered that she had a very serious and relatively rare heart condition known as hypoplastic left heart syndrome, or HLHS. In this syndrome, the dominant feature is the small left ventricle. There are many causes for this. Many reasons why this might developmentally happen. In our daughter's case, her mitral valve was stenosed. It wasn't working at all, and so no blood was getting through to the left side of her heart. In other cases, the cell wall may be underdeveloped. These babies are born apparently healthy because of this ductus arteriosus, a connection, a hole in the heart, between the aorta and the pulmonary vein. Developmentally, this allows the lungs to be bypassed during the fetal period because the baby's not breathing, doesn't need to be getting oxygen through the lungs, but the lungs still need to be able to develop and have blood going through them so that they're developing properly. So there's this hole in the heart to bypass the lungs, to decrease the pressure on the lungs, and to make it so that the oxygenated blood that is coming from the umbilical cord is actually being pumped to the rest of the body. So these babies, they have this hole in their heart. All babies have it, and so they have some oxygenated blood that is getting out into their heart from their lungs and then being pumped to their body. This hole closes up normally. That's a part of the early neonatal period. The hole closes on all babies between 2 and 5 days old. And then at that point, babies with HLHS no longer have a connection between their body and their lungs, and therefore their lungs are not able to deliver oxygenated blood to the body. There are some treatments for this. Obviously, we did not get into that, but they have some heart surgeries that they can do. The first one has to be done within the first week of life that reroute the heart, the veins and vessels in the heart, so that oxygenated blood is then being pumped by the other chambers of the heart. It is dangerous, obviously. Doing heart surgery in the first week of life is very dangerous. But the mortality rate is going way down. I actually currently know a little girl. She's my son's age, so she's 9 or 10. She's doing well. She's had to have a heart transplant because of complications, but she's doing really well. So it used to be a death sentence, 50% mortality each time you had a surgery, and so most kids didn't live past age 5, and now they have people that are in their early 20s that are living with this heart defect. So I was interested in this. I was thinking that I might want to research and see what is known about the causes, genetic or developmental, of hypoplastic left heart syndrome. But what I found was there's not really a lot known. It seems to be a very multivariant disease presentation, and there doesn't seem to be any known common or even set of common factors that lead to this disease. However, I did find an interesting article. This was a case study where they found a defect in PKP2, or plakophilin 2. This was kind of a sad story. This couple from the Netherlands had 2 children that died in utero and were found to have hypoplastic left heart syndrome. as well as other defects. And upon genetic analysis, they found this defect in this protein, PKP2. PKP2 is a protein, a small protein, that is involved in desmosome structure and function. Desmosomes, as you know, are involved in cell-to-cell adhesion. And the PKP2 is especially necessary in cardiocytes, which, as you can imagine, need to withstand a lot of stress over their lifetime to keep the heart muscle functioning and the body alive. PKP2 specifically links cadherins to the intermediate filaments within the cell. Recent research has also shown that they are involved in cell-to-cell signaling within the cardiocytes. That this part is not well understood yet, but something about the way they work involves them in signal transduction between cardiac cells, and therefore it's the defects in plakophilin 2 are also implicated in arrhythmias. As I did a little bit more research on this particular defect, it was not shown that it was specific to HLHS, but that it is implicated in 40 to 50% of cases of arrhythmogenic right ventricular dysplasia. Now, you may have never heard this name and don't know anything about this cardiac defect, but I'm pretty sure you have heard news stories about it because it is the most common cause of sudden cardiac death in young adulthood, particularly in young athletes. So this would be the most common cause of when you read a news story that says 19-year-old football player falls dead during training, or a basketball player fell down while running around the court. 40 to 50% of these sudden cardiac deaths are due to this particular disorder, which is most likely caused by PKP2 mutation. This mutation leads to defective cardiodesmosomes, which means that the cells are not able to connect to each other as well. They aren't able to withstand the stress of the cardiac muscle pumping. Under normal conditions, early in life, the first 10, 20 years of life, there are no problems. The cardiac muscle works well enough for everyday life, and these individuals do not suffer any symptoms. But over these years, slowly their cells are losing connection and then dying and being replaced with fibrofatty tissue. As you can see over here, the upper slide is showing a picture of normal cardiac tissue, and the lower slide is showing damaged cardiac tissue that is being replaced with these fatty deposits and with fibrous tissue. Eventually, this leads to cardiac muscle, the whole muscle, that is just not as strong as it used to be, and therefore is not able to withstand the rigors of athletic training specifically. Some people with this condition do live longer, especially if they were not a student athlete, but eventually their cardiocytes will not be able to hold up. There won't be enough cardiac muscle cells left for their cardiac muscle to work very well. An interesting study that wasn't directly related to PKP2, some researchers used some advanced electron microscopy techniques coupled with some space-filling models from a computer program. And they just happened to use the mutated PKP as their, not control, as their, what they compared the normal cardiac cell slides to. Over here on the left, they're showing a normal cardiac cell. The computer program has put in this green ribbon, which is a space-filling model. It's not an actual structure in the cell. It's just showing how much space there is between these cardiac cells. And it's showing that this is a normal amount of space between the cardiac cells. Over here is the cardiac cells with the mutated PKP2 molecule where the desmosomes are not as structurally sound. And the space filling model shows that the space between these cardiac cells is much wider, and the cells are just not as attached. They are not able to hold on as well. Heart development starts very early on with the development of the first and second heart fields. The heart fields come from cells that migrate from the splenic mesoderm in the lateral plate. They have been specified to be heart cells, and they travel to the thorax where they form a tube. Over here, you can see this is the first heart field. This is after it has formed what is known as the cardiac crescent, which is just a tube. It is the very early heart stage, a very primitive heart. Some organisms on this planet that's where their heart development stops, is just in a tube. The second heart field is just behind the first heart field. It is also the same specified heart cells that travel, and eventually the first heart field will become the left ventricle, and the second heart field will become all the right and left atrium, the right ventricle, and the outflow tract, so the aorta and the pulmonary veins. This picture shows the same developmental process. The cells migrate to the cardiac area where your heart is going to be. On either side, they form a tube. Each side forms a tube, and the tubes fuse into the primitive heart tube. This tube then starts to bulge outward a little bit and turn, and this becomes the left ventricle. Here is the right ventricle, and the atria form behind it. It is interesting to me how many steps go into forming the heart, and it's not surprising that there are so many defects that can happen throughout this process. Most heart defects are very minor, a little hole in the heart, a little murmur, but some heart defects are extremely serious, including not forming parts of the heart or having parts that are attached in the wrong way, transposition of the great vessels, for instance. Many kinds of heart defects are developmental in nature, where there's just something that went wrong with how all of these developmental steps, and something went wrong, and one step was missed or done incorrectly. Of interesting note is that all the cells in the heart, myocardium, epicardium, endocardium, and the Purkinje fibers, all form from these heart fields, which come from the splenic mesoderm. So the heart is not formed by 3 layers of different kinds of cells coming together and making the chambers. The heart is formed from one cell type that differentiates into all the cell types that the heart needs. This means that the very early heart cells are pluripotent and can become any type of cell. One of the first papers that I found is called "The Molecular Disturbance Underlies the Erythromyogenic Cardiomyopathy." They are using a mouse model to study this heart defect that causes this sudden cardiac death in young adults. They developed a mouse model, a transgene mouse model, that had the truncated PKP2 molecule. They created 5 transgene lines that had varying lengths of PKP2. So some of them were much closer to normal, and some of them were very short. And they found that the shorter the PKP2 model, the less stable the PKP2 molecule was, and therefore less stable the desmosome was. In this picture from this paper, over here they are showing, this is the cardiac muscle that is in the transgene mouse, and this is showing the intercalated disc and how wide it is in this mutated mouse. The desmosomes are just not able to hold it together enough for it to be normal. In this study, they also found that damage to the cardiac muscle was age and exercise dependent, meaning that the older the mouse was, or the more the mouse exercised, the more cardiac damage they sustained. Now this makes sense. If the desmosome is less stable, the more you stress the heart muscle, either through age, the more they had lived, the more the heart muscle has worked, or through exercise. Both of those resulted in more heart damage over time. And it was also dependent on how short the PKP2 molecule that they were using. If it was the shorter one, the earlier they saw the cardiac abnormalities. So this fits in with what we know about the sudden cardiac death in these young athletes. They've lived 20 years or so, and slowly their heart has become less and less able to withstand the stress of daily life, and they put themselves through a intensive football regimen, for instance, and it proves to be too much for their weakened cardiac muscle. The final paper that I found was suggesting a treatment for this condition. They had found that the adenosine 2-alpha receptor was involved in intracellular signaling that led to this profibrotic changes, this fibrofatty deposits that replaced the cardiocytes. And so they used a mouse model and blocked this adenosine 2A receptor, so that's what this Istra is. That's the medication that they gave that blocked this pathway that was leading to fibrosis. And they found that up to a certain point, it protected heart function. On this side, they're showing the untreated mouse heart and how it has a lot of fibrosis in it. And here is the heart muscle that has been treated with Istra, and it doesn't have as much fibrosis. Over here is the chart that shows percent collagen. So here's the right ventricle, here's the left ventricle. The red is the untreated mouse heart, and the black is the Istra-treated mouse heart. And they are showing that the Istra protected the heart from laying down more collagen. The right ventricle of the Istra-treated mouse had 40% collagen. The untreated mouse had 60% collagen. Now they had a couple of caveats, I guess, for this. One is that the mouse model may not translate into the human, so this isn't necessarily proof that there is a treatment for this condition in humans. And number two, this is not a cure. It is a short-term, relatively short-term treatment to protect what function is there, but it doesn't protect it long-term. So they suggest that this proposes a treatment that could be used by people who have this arrhythmogenic right ventricular cardiomyopathy. If they're starting to have symptoms, if they're waiting for heart transplant and they need their heart just to last a little bit longer. But it is a very interesting idea that blocking a receptor that doesn't necessarily seem like it's related to PKP2 can help protect the heart from further damage. I hope you enjoyed this presentation and learned something about how the heart works and how various kinds of defects can affect cardiac development and cardiac defects. Thank you.